This summer I worked with the NSF RU program examining patterns of Dioicy across the Hornwort phylogeny using UV sex chromosomes. Hornworts are a lineage of bryophytes, which is the deepest diverging clade in land plants. Bryophytes occur in a haploid dominant phase, which is where each chromosome only has one set, or there's only one chromosome. So that means that in hornworts that have sex chromosomes, they have UV sex chromosomes. Because they occur in only singularity, the UV sex chromosomes won't degenerate, because natural selection occurs on both chromosomes equally, as opposed to the degeneration of the X and Y chromosomes. This is an example of a monoecious hornwort, which is a hornwort with both the male structure and the female structure on the same plant, whereas a dioicous hornwort has them on separate plants. 40% of the hornwort phylogeny is dioicous, with many different independent events of evolution from monoicy to dioicy, and then one example of dioicy to monoicy. The goals of the study are to determine if the samples are male or female, identify sex chromosome structure, identify any important genes, and then do some inter- and intra-species comparison. These colors here coordinate to the colors here, and what we were able to accomplish. We were unable to accomplish nanopore min-ion sequencing, so we did Illumina whole genome sequencing, and then we almost got through the RNA sequencing pipeline, but we ran out of time. Down here, these graphs show the difference between the reference genome and the sex chromosome, with less difference being along these lines, and then more difference diverging from that line. So on the autosome, or the non-sex chromosome of a same-sex example of Phylogeny, it's very similar to the reference genome, and the opposite sex, an autosome, is also very similar to the reference genome, whereas the sex chromosome for the same sex sample is the same, and then the sex chromosome of the opposite sex sample is very different. The same thing, same type of graph down here, where the difference between the reference and the sample genome is plotted. The area that has less of a difference from the reference genome, we think, is the pseudo-autosomal region of the chromosome, or the area on the chromosome that can still recombine with the other sex chromosome. This is a different type of graph with different type of data that correlates to that pseudo-autosomal region in Leosporoceros. The dark green line means a one-to-one comparison between the sample and the reference genome, whereas this kind of broken line here is where it's not plotting as well, and that would be the sex-determining region of the sex chromosome. The same thing is true over here with Phaeomegaceros and Phymatoceros, where Phaeomegaceros has 3 pseudo-autosomal regions and 2 sex-determining regions, and Phymatoceros just has a sex-determining region. And then this is the whole genome type of graph, where the dark green line is a one-to-one comparison along the autosomes, and then where it can get kind of jumbled, as we can see on the Phaeomegaceros, is the sex chromosome. These are some statistics for the reference genome, the sample genome, and then the RNA data for Phymatoceros, Phaeomegaceros, and Leosporoceros, which tells us about the total genome size, how fragmented the genome is, and then how complete the genome is. Overall, I was able to complete 2 out of my 4 goals, with being able to identify important genes not getting completed because I ran out of time, and then without identifying important genes, I was unable to do any inter- or intra-species comparison. So the next steps would be using OrthoFinder 2 to identify important genes and compare those genes between different species. Overall, I'd like to thank Peter Schafferin and Fei Wei Li for this summer.